Hello fellow humans, my name is Nikki and I am a scientist on a mission. I did my undergrad in biochemistry and I'm doing a master's in microbiology at the moment. And my mission is to teach you about the different pathogens in the world. In today's video, I wanted to go through and teach you about the Ebola virus following the outbreak that occurred from 2018 to 2019. I'm going to break up the video into different sections. I'm talking about different parts of the virus, the diagnostics, the surveillance, the treatment plans, and the preventative methods that we've got in store. So let's get started. So part 1, the virus. So the virus and the morphology looks like this. Isn't it really cool? It's super cool. So it has a morphology that I've actually never seen before in virology, and it's very, very long. And I'll show you just here. This is a picture that shows a heavily infected monkey kidney cell that's infected by Ebola, which is the ones in the green, right? So it is very, very, very virulent. How does it actually work? Now that we know it looks like this, how does the virus actually get into our cell and become as dangerous and as pathogenic as we know it to be? You have immune cells called monocytes and macrophages, and they are one of the first respondents in an infection. So Ebola itself, the virus infects these cells, it infects these immune cells, and it goes into the cell, binds to the receptor, and once it's taken inside the cell, it actually fuses with a protein on our cell, and a protein called NPC1. So it's essentially a fusion protein that allows the virus to punch a hole into the lysosome, that vesicle that it's in, and it secretes its DNA out into the cytoplasm, out where all of our DNA replication mechanisms are. So once it's inside, it replicates its viral genome using our own machinery, and this is a very parasitic function of viruses. They cannot replicate on their own, so it replicates using our genome, and it puts itself together. So one of these viruses going in replicates itself and releases millions of other Ebola viruses out into neighboring cells and infects more cells, and goes up and infects the brain, the kidney, the organs, the lungs, the heart. It can infect all types of cells. It's not contained within a one specific area. It goes and spreads across the entire body. Huge inflammatory response and a huge immune suppressant, and the loss of ability to actually respond to the virus in part as the symptoms start to progress and it infects further and further. So part 2, this is the symptoms and disease. So the Ebola virus is quite often known to cause a very fatal severe symptoms in both humans and primates. At the very beginning, you have when you contract the Ebola, you have very mild symptoms like fever, fatigue, headaches, sore throats, muscle pains are often characteristics of the Ebola virus. As it starts to get more severe, there's vomiting, there's diarrhea, there's impaired kidney function and liver functions, and both internal and external bleeding. One of the issues with diagnosing Ebola in the very beginning is that its symptoms, so both fever, fatigue, muscle pains, and stuff like that. So they are symptoms of other types of infectious diseases like malaria, typhoid, and interestingly as well, there are also symptoms that are noticeable in pregnancy. So it just means that when you go into rural places, it's really hard initially to let the patient know that you suspect that they have Ebola. So, part 3, the diagnostics and surveillance. So we actually have a lot of ways of testing whether or not a patient tests positives for Ebola. These are the different types of techniques that we can use to get a positive result, but a lot of these techniques are laboratory based. So like I mentioned before, we don't actually have any ways of testing for Ebola with the patient at the time. So the World Health Organization recommends 2 different types of samples that you should get from the patients or suspected patients in order to confirm whether or not they carry the Ebola virus. So sample 1 that you can get is white blood cell samples. From there you do those tests that I mentioned before, or sample 2 in the case where you can't get blood samples or deceased patients, it's recommended that you require salival samples. Unfortunately, the way in which the virus spreads is through bodily fluid, so it just means that the samples that are taken by healthcare workers are really high biohazard samples, and must be treated with care. So, part 4, the transmission and epidemiology. So, historically, Ebola outbreaks have been localized in rural areas in Central Africa, and it was first isolated in 1976. Our first assumptions with these smaller viruses is that it was initially a zoonotic transfer, so this is from animals to humans, and this can happen in a number of ways. So, either through the consumption of raw infected meat of these animals, or through biting and abrasions, and the virus can enter the skin that way. So, following the 2014 Ebola outbreak, we have realized that human-to-human transmission can occur as well. So, in a lot of cases, the Ebola virus actually is secreted in a lot of your bodily fluids. So, people can get infected through blood samples, through urine samples, through other body secretions. Pregnant women as well have also been found to test positive with Ebola in their breast milk, and there's also sexual transmission that occurs as well. So one of the most notable cases was the 2014 Ebola outbreak, and that was the case that sort of drew everybody in prior to, after the 80s and after the 90s. So the severity of the 2014 West African Ebola outbreak is actually quite uncharacteristic, and it isn't something that we had expected or were able to prepare for. The largest outbreak of Ebola virus in our history occurred between March 2014 and June 2016, and this was an outbreak between 3 countries, and this was going from Guinea and spreading down to Libya and to Sierra Leone. So in this outbreak, it infected more than 28,600 people, and there was an estimated 11,000 deaths during this. So you can see in this graph here, I've characterized the number of cases and the overall death. So in Sierra Leone, there was a fatality rate of about 22%, going down to 67% in Guinea, which was the highest at the time. So after 2014, there's actually been another major outbreak of Ebola occurring in around June to August of 2018, and is ongoing currently in the Democratic Republic of Congo. So as of February 2019, there has been confirmed 3,432 cases with 2,249 deaths reported in the country. So the World Health Organization actually declared this outbreak a public health emergency of international concern. This means that the international community was forced to respond, and a lot more funding could be allocated to controlling the spread. So, part 5, prevention. So the World Health Organization has actually defined a 3-step plan on how we should control the epidemic. So just 3 steps. What are they? So number 1, we need to be able to isolate the patients and confirmed cases in order to prevent further transmissions. Number 2, we need to be able to recognize new cases as they arrive and contact the relatives and friends of the person who is suspected to have Ebola, and monitor them for the next couple of days and bring them in should symptoms arise. And number 3, perform accurate and respectable burial procedures in order to contain spread through other bodily fluids excreted from death. So just 3 steps. There's different approaches that we can use in order to try and go about this. So number 1, we need to contribute to community engagement, and have healthcare workers who speak the native language and understand the customs, and come from that rural area, engaging with the community rather than foreigners. Because if you have this guy coming up into your doorstep and saying, hey, you have Ebola and we're going to take you away from your community, or hey, your mother is very sick, but you can't touch her and you can't go in there, and he looks like this, you're probably not going to listen to him. So that's one of the ways that we're trying to go about it, is we're trying to have healthcare workers who already exist within the communities, the rural communities, who understand the language, understand the customs, to go in and talk to them. One of the things that prevents us from going about this procedure is there's quite a lot of reasons. So the first thing is the actual nature of the Ebola virus itself. It is very unpredictable, and outbreaks themselves are very sporadic. It's hard to quantify how large the outbreak will actually be when it occurs, and there's multiple different strains involved in each of the different infections in each of the different countries, so it's hard to watch them all. Another thing that inhibits it as well, and one of the major things in the Democratic Republic of Congo itself, is the actual inter-ethnical violence that occurs and the political instability within the country. So it's hard to go in and try and actually intervene with all the violence that is currently occurring. One of the limiting factors that you might not have considered is the marketability of treatments, or the marketability of vaccines. It just means that the necessity of the Ebola vaccine remains largely unpredictable, and the quantities in which we need to use them remain unpredictable as well. So if you're a company that's developing Ebola vaccines, you're developing it for a clientele. Most of your clientele is probably going to be government and non-government agencies that are going to be the sole buyers, and they're going to be redistributing it to rural communities. So it's hard to know what quantities you'll be using, and then it's hard to also invest in years and years of Ebola research. You know, if you go from 31,000 infectants to like 20 infectants in the following year. So part 6. Treatment. So now that I've told you all the loom and gloom about the virus, and the way that it works, the way that it infects, and the death rates, how do we actually go about controlling it? So we don't currently actually have any treatments for Ebola, which is quite sad. If we can identify that an infected patient very early on, we can go about treating their symptoms. So one of the largest killers of Ebola is the dehydration. So you're vomiting, you have extreme diarrhea, you're excreting bodily fluids, you're sweating all over the place. Each of these isn't containing viruses and being able to spread should a person have contact to them. If we can alleviate a lot of the effects of the symptoms, so rehydration, we can detect it very early on before it gets very serious. There's actually a really high survivability rate. A vaccine that has just been licensed and is coming out, and this, together with the World Health Organization and Gavi, which is the Vaccine Alliance, about and gotten pre-licenses to these vaccines, and then they've dealt an agreement, and I'll show it to you here. So agreement between Gavi and Malbec, which is the developer for the vaccine, that ensures emergency stockpile of about 300,000 investigative doses in the event of an outbreak. Following the success of the trial, once the outbreak had occurred in 2018 to 2019, we have successfully protected about 250,000 people, and it's proven quite effective after a single dosage in preventing the disease. Now that you've sat through the entire video and you've learned all there is to know about the Ebola virus. So people are getting better from Ebola. It's not a death sentence that we seem to think it is. The high death rates occur in rural areas where they don't have access to medical facilities, they don't have access to hospitals and stuff like that. So yes, can I help you? Ignore her. Cool. So now you know everything that we know about the Ebola virus. So thank you so much for getting this far, and I really hope that you learned something and were able to take something away from this, and were excited as I was, staying up at 2 a.m. just doing a bunch of reading, just excited to learn about the virus itself. And I hope that I've been able to, you know, get you as excited about the type of research that's coming out in for Ebola in the future. Yeah, so if you have any other viruses that you'd love to learn about, or bacteria, I am a bacteriologist as well. If you have anything that you want to learn about, shoot me a message down in the video, or if you have any questions about the Ebola vaccine, comment as well, and I'll do my best to answer all of your questions. And I will have all of the references, all the information on this website here, and I've got it in the link as well, and you can read the blog that's relevant to the video with all my references, including the World Health Organization and Gavi as well, and the work that they do with the outbreaks as well, phenomenal research that they do, and all of, you know, a large thank you to all the healthcare workers that can work as first respondents in cases like this and deal with the, you know, the backlash within rural communities and put their lives on the line every single day for something like this as well. So I hope you grow a larger appreciation for what it is that they're doing and what they are trying to achieve. So if you liked it, please comment and subscribe below for more videos like this. I will aim and do my very, very best to produce a video every single Sunday and once a week. So I hope you have a lovely day.